Hello everyone, I'm Aurora Bakaj from the Biology Department, University of Vlora, Albania. Me and my colleague have been working on water analysis, especially river, lagoons, and also seawater analysis, as Vlora region is very rich with lagoons. It has two lagoons, Sorikumi and Narta Lagoon, and also has many rivers coming across the region. And also it's a beautiful city as it is a seaside city. I'm here today to present a work done in Narta Lagoon with my colleagues. Evaluation of environmental situation in Narta Lagoon, Vlora, Albania. These are colleagues that have been working on this project. The aim of the study is the assessment of environmental condition of Narta Lagoon, as well as the analysis of the impact of anthropogenic activity based on comprehensive analysis with specific reference to the value of microbiological indicators and physicochemical parameters of water lagoon. Introduction. Narta Lagoon is the second largest coastal lagoon in Albania. It is separated by a narrow littoral cordon with the sea, with Adriatic Sea. Due to its position between the land and the sea, it is a highly productive coastal ecosystem. Narta Lagoon serves as a refuge and food area for many continental and marine species. Coastal lagoons are exposed to significant anthropogenic pollutions from both point and non-point source. They are also subject to high anthropogenic pressure that comes as a result of increased human activity, urbanization, and industrialization. Factors such as waste discharge from various sources, from agriculture runoff, domestic and industrial runoff, industrial waste, untreated wastewater, various waste during boat ballast, uncontrolled fishing are responsible not only for chemical and biological deterioration, but also for changes in the microbial ecology of water in these areas. Increased concentration of microbial indications of fecal contamination originating from different sources may be an indication of high risk, such as the presence of other pathogenic microorganisms, pathogenic bacteria, enteric viruses, or even other parasites. These waters can consequently lead to the transmission of pathogenic and waterborne disease. It is therefore very necessary to understand the extent of fecal contamination. It is important to carry out continuous monitoring of microbiological indicators such as total coliform and fecal coliform bacteria, as they will shed light on sanitary status of recreational water, fishing purpose, etc. Material and methods. The study area. Here we have the study area. Here is Albania, and we have zoomed in the study area. You may see here Vlora region. And here you have it zoomed in, and now here is the region where is situated Narta Lagoon. Narta Lagoon has a surface approximately to 45 kilometers square. It is a shallow lagoon with an average depth of 1.2 meters. It is located in the north of Vlora city. As you may see, here is Vlora city. The lagoon is bordered on the north of the saline through a dam. On the southern part, it is bordered by the land, as you may see, and in the west it is bordered by the Adriatic Sea. Between the lagoon and the Adriatic Sea, there is a littoral cordon mostly covered by pine forest. Sampling station. Here you have a map of the sampling station and also their coordinates. This study was conducted during a 12-month period starting from December 2017 to November 2018 in 4 different seasons: winter from December 2017 to February 2018, spring from March to May, summer from June to August, and autumn from September to November. Water samples were collected in 7 different stations distributing along the lagoon perimeter, starting from the first connection point with the sea. Here in this map you may understand that there are 2 channels in which Narta Lagoon communicate with the sea. Is the first channel here when we took the first sampling, and for this reason it is called the first sample station. The other is here, the second sample station. Here is Zvernetsi Island, and near the Zvernetsi Island we have the third sampling station. The fourth sampling station, as you may see here, is near a discharge channel that comes from Narta town. And near the highway, here is the highway, near the highway we have the 5th and the 6th sample station. In the north of Narta Lagoon we have the saline Scrofotina, and here at the end we have the 7th sample, the last one. So, as I mentioned before, the first sample station is the first connection point with the sea. The second is the second connection. The third is near Zvernetsi Monastery, which is located in the Zvernetsi Island. The fourth is situated on the north of Narta village, near the irrigation canal. The fifth is situation in the northeastern part of Narta Lagoon, located near the highway, and the 6th and the 7th sample station are situated in the north part of the lagoon, as you may see here, and along the Scrofotina barrier. We have the parameters that we analyzed. So in this study we have been dealing with physical-chemical parameters as well as microbiological parameters. For physical-chemical characteristics of water lagoon, we used samples taken with glass bottles. For analyzing these samples, we use the methods described by APA and other types of standards. Temperature, pH, and salinity were the physical-chemical parameters analyzed. Water temperature was taken in situ by placing the thermometer at the depth of 10 centimeters, while pH and salinity were measured in the laboratory. Sampling technique for microbiological analysis. The sample were collecting from the surface water 10 to 30 centimeter at each site using 1 liter sterilized glass bottle. The collected water sample were transported to the laboratory in thermal boxes within 2 hours. Lab work was performed based on standards at the Microbiological Laboratory of Biology Department, University of Vlora. During the sampling was recorded the exact time and date, water temperature, and some data related to weather condition. Biological analysis and treatment of water sample were performed according the regulation and standards. Throughout the monitoring period, we collected 336 water samples and we analyzed them according standards. Microbiological analyses were performed, as I mentioned before, in the Microbiological Lab of Biology Department, University of Vlora, referred to different standards. Determination of total and fecal coliform was performed according to the MPN method using the combination of 333 tubes. So here we have 6 tubes. There are many others. In fact, for each sample we have 9 tubes. That was used was at a broth. The diluted sample were incubated at 35 degree for the preliminary test and 44 degree for the confirmation test. We read the analysis at 24 and 48 hours. Bronchresol acid purple broth was used in confirmation tests. And the data was expressed significantly with the number of organisms in CF. Statistical analysis, seasonal differences between the sampling locations in terms of bacterial levels and environmental factors were evidenced in box plots and mean plots of fecal coliform distribution. The comparison of fecal coliform values at different sample stations and different months was performed using one-way analysis of variance ANOVA. When a significant difference for the main effect was observed, a Turkey pairwise comparison test was also performed. The correlation between the bacterial levels and the factors were determined using Spearman correlation analysis in SPSS. Result and discussion. The aim of our study, as mentioned before, was the evaluation of lagoon water quality based on comprehensive analysis with specific reference to values of microbiological indicators and physicochemical factors of water lagoon. For a completed pattern, we analyzed 7 different sample stations eventually distributed along the lagoon perimeter. Description analysis of all sampling stations. We may notice that the value are stable for the first 3 sampling station with a low variance, which indicate a low environmental impact in all these sample stations. The data obtained from the 4th to the 7th sampling station characterized by a high coefficient of squamous and positive coefficient of kurtosis. So our data set are slightly skewed right and tend to have heavy tails. Based on the above statement, fecal indicators are influenced by the time condition of sampling. A clear representative of the obtained data shows that the total number of coliform bacteria is highest in sampling station 4, 5, 6 and 7, with a minimum value of 23 and in winter and spring and 2,400 in summer. Here you have 2 different graphs. In the first one there are the value of coliform bacteria in winter and spring, and in the second in summer and autumn. As you may see here, it's clearly stated that the value are higher in the springtime, probably because of the raining and as well as the runoff from the agricultural work. And also we have high value in summertime. These values are probably because of the high temperature and the lower of the level of water in the lagoon and other factors analyzed. So based on the box plot of microbial distribution in different sampling stations as well as in different months, we can say that there are many outliers present in our data set, especially in sampling station 4, 6, 5 and 7, and you may see here. So in sample station 4, 5, 6, 7 we have outliers, and we may understand that these outliers are in these months, are in spring and in summer. Based on the mean plot of microbiological distribution in different sampling stations, as we may see here, and also in different months as it is here, we may notice a complete different situation that occurs at sample station 4. You may see the sample station 4 has a high value of coliform bacteria. This station is located in the northeast of Narta village near the drainage canal that flows into the lagoon. This irrigation canal has a big influence in the fecal contamination of the lagoon. Additionally, there was a statistically significant difference between stations in terms of total and fecal coliforms, and also between months. ANOVA results revealed that the variation between the sampling stations were significantly in terms of fecal coliform levels, also for different months. This result was checked with the post hoc Turkey and other tests. These variation results are from the high levels of contamination present in sampling station 4 near the drainage channel. Moreover, Spearman's correlation coefficient matrices were calculated for fecal coliform month and temperature. The relationship between physical parameters and coliform levels were statistically analyzed because of fecal coliform counts are affected by several environmental factors. The rate of die-off increases with increasing of temperature, elevated pH, higher oxygen levels, and lack of salinity. The results show that coliform bacteria were correlated with the temperature and also with pH and salinity. As a conclusion, in this study, we focused in the lagoon water quality expressed by indicator bacteria and physicochemical parameters in concentration levels in water sample. We selected the sample station in order to cover all the hydrodynamical characteristics of the lagoon. Human impact on the quality of Narta lagoon water was more than evident. Based on our data, the most polluted sample station was sample station 4, as it is located near the drainage canal. Sample station 5 and 6 were also polluted as they are located near sample station 4. According to Zhu et al., it is well known that fecal bacteria sources explain direct discharges from rivers, channels, and storms. Narta Lagoon is a lagoon of high ecological and economical importance due to the biodiversity that it presents. pollution of the lagoon due to rains, urban runoff from the drainage canal, agriculture runoff, fishing activity, increased human activity, fetches of living organism, and water wolf will affect human and aquatic life in the future. Therefore, we suggest that periodic study of the environmental condition of the lagoon should be carried out. Here there are some references of the study, and thank you very much for your attention.